Hi, today I'm going to talk to you about the project that I've been working on, and this is the exercise intervention in an obese mouse pregnancy. So this work has been done at the University of Cambridge in the Wellcome Trust MRC Institute of Metabolic Sciences and under the principal investigator, Professor Sue Ozanne, and has been funded by the British Heart Foundation. So why are we interested in obesity? Well, obesity in pregnancy has a significant impact on the woman, on the fetus, and also on the long-term health of the child. Specifically, offspring who are born to obese mothers have an increased risk for obesity, diabetes, and cardiovascular disease, which cannot wholly be accounted for by an increased genetic inheritance or their current environment. So we are particularly interested in looking at obesity in pregnancy on the impact of the child. So the aim of this study was to look to see whether an exercise intervention in an obese pregnancy could reduce the risk of cardiovascular disease in the offspring. So in order to do this, we have three groups. We have our lean control mice, we have our obese mice, and our obese exercised mice. So these obese exercised mice undergo a treadmill training regime, which is not voluntary, and then we aim to look at the offspring at 8 weeks of age. And so in terms of their cardiac structure, we are able to look at using histology, cardiomyocyte cell area again using histology, and we will also look at their cardiac function, which is done by echocardiography, and non-invasive blood pressure measurements, which is done by a tail cuff method. So before I go into detail about the study and the results, I thought, being as everything is virtual, that I ought to introduce myself to you. And so I'm Heather, and I'm a postdoc with Sue Ozanne. Here's a picture of me in early lockdown in the UK, working from home around children's toys. But I started off my academic life doing my undergraduate degree at Durham University in biomedical sciences. I then moved to Cambridge, where I then stayed. I was one of the first students on the British Heart Foundation 1+3 Masters PhD programme, and I did my PhD with Professor Sue Ozanne, looking at cardiovascular effects of maternal obesity. And then now I'm a postdoc, again still with Sue, and funded by the British Heart Foundation, working on a number of projects, including this one that I'm going to present to you today, which also formed a large part of Dr. Jessica Beeson's PhD when she was in the lab. So one of the things that I enjoy doing is sewing, and I guess since lockdown, my priorities have gone from my daughter's dresses to now face coverings. So this is an interesting change. So now I've introduced myself to you, I want to introduce you to the mice and the groups that we will be using throughout this presentation. So I have my first group, which is the control group, and these mice are C57 black 6s, and they are fed a controlled chow diet from their weaning throughout their lives, so pre-pregnancy, during pregnancy, and lactation. And then when the offspring are weaned, which is around 3 weeks of age, they are also weaned onto a chow RM1 diet. And the studies that I'll be presenting to you today will show the offspring at 8 weeks of age, where we do a post-mortem. And just before this, the cardiovascular analyses are performed. So moving on, we have the obese female mice now, and these females are fed a high-fat, high-sugar diet from their weaning at 3 weeks of age throughout their lives, so pre-pregnancy, during pregnancy, and lactation. So these are high-fat diet pellets, and they also have sweetened condensed milk. in a pot in their cages. So what is really important to note is that in these females, they will not be mated for pregnancy until they reach at least 10 grams of fat mass. And this is to ensure that they are truly obese when they go into pregnancy, and they will be at least double the fat mass of these control mice here. And so again, what is important to note is that the offspring are weaned onto a standard laboratory chow, so this RM1 diet here. And this is important as it highlights that any differences that you see in the offspring are a result of the differences here in the maternal diet. So our third group is the obese exercise group. So what is important to note here is that these mice are undergo exactly the same dietary conditions as the obese group, with their only difference being that they are put on a training regime just before pregnancy and during pregnancy up until E17. So we initially start 1 week before mating, and they are trained on an exercise treadmill where we increase the speed across that training week just to get them used to being handled and get them used to the treadmill. Then they are mated with the males and they're no longer exercised, and then once they plug, they are then exercised 5 days a week at a consistent speed of 12.5 meters per minute. So this exercise training regime is not exhaustive, it is moderate, and we don't wear the mice out at all. And so what is important to note here is that by E17 we stop the exercise regime just before birth, and these mums are maintained on this high fat, high sugar diet all the way through up until lactation, where the offspring again are weaned onto a standard laboratory chow. So what the take-home message really from this slide is that the offspring, so here, are fed a standard laboratory chow, and that any differences that we see is down to the differences in the maternal environment. So I'm going to start off by talking about what we already know in terms of the heart phenotype from offspring of control mothers and offspring of obese mothers. And then later on, I will add in the exercise group. So what do we already know? So there's several studies that we've published over the years in this model looking at the heart, and we have shown that at 8 weeks of age that these offspring develop cardiac hypertrophy. They have an increase in heart weight relative to body weight and also to tibial length, and this is also coupled to an increase in cardiomyocyte cell size. These offspring were also shown to be hyperinsulinemic and very sensitive to insulin, specifically in the heart, but not necessarily the other tissues. So we believe that the cardiac hypertrophy may be driven by these offspring being hyperinsulinemic. So as part of my PhD, I looked at an isolated Langendorf heart perfusion model, which is where you take the heart from the mouse and you mount it onto a cannula and perfuse it with oxygen and a Krebs solution, and then you look at baseline cardiac function in the left ventricle, and then you do a stimulated response using various drugs to look at sympathetic tone. So what we showed here, and this is in 12-week offspring, so with the obese offspring in the black bars, we showed a reduction in cardiac systolic function here, shown in A and B, with a reduction in the left ventricular pressure and a reduction in the ability to contract. In addition to this, we showed that the offspring of the obese dams, again in black in C and D, had an impaired diastolic function with an increase in the left ventricular end diastolic pressure and a reduction almost significant in the ability to relax. So when we looked in the stimulated environment, where we added isoprenaline, which is a beta-1 adrenergic agonist, and also carbachol, which is an M2 muscarinic agonist, we showed that in terms of the heart rate response and also the contractile response, that these offspring of obese dams were very highly sensitive to sympathetic stimuli, and hence are termed as sympathetic dominant. And so this, whilst initially it is a compensatory mechanism that would be adopted to increase cardiac function and cardiac output, long-term hyperactivation of the sympathetic nervous system is not a good thing and is often associated with phenotypes in humans such as heart failure. So I want to start off introducing the treadmill part of the exercise regime and look at what it does to the mother, as this is important when you think about what the effect is on the offspring. So to start off, I am showing you maternal body weight, and as you can see here, we have the control groups in green, and they are a nice healthy 25 grams, and they increase their body weight over pregnancy, which is what we would expect. This is also the case in the obese exercise group, except they are significantly heavier than the controls. And what is important to note here is that the offspring of the obese exercise group do not lose weight by exercising during the before pregnancy and also during the pregnancy period. So we also did fat mass analysis using TDNMR, and as you can see here, there is no significant differences between the obese and the obese exercise intervention group in terms of their fat mass. So I guess the take-home message from this slide, and what I want you to take through the presentation, is that these mothers do not lose weight or fat mass on their exercise regime, that it is a moderate exercise regime. So in addition to the body weight data, we've published data showing that actually that this moderate exercise with a lack of effect on body weight or fat mass is capable still of improving glucose tolerance and also reducing the hyperinsulinemia that would otherwise have been seen with the dams just being fed an obesogenic diet. So what impact does this maternal intervention strategy have on the metabolic health of the offspring? So this is something that I'm just going to lightly touch over, as I'm more interested in what the cardiac phenotype of these offspring are, but I thought that you may be interested in this nonetheless. So at 8 weeks of age, which is when we did our post-mortem, we showed that there were no differences in the offspring, now we're talking about, in their fed body weight. So as you can see here, they're both around, all three groups are around 25 grams. What we did notice was that there was a borderline significant increase in fat mass in the offspring of the obese mothers, but actually that this was corrected by the exercise intervention, where levels were brought down closer to that of the controls. There appeared to be no changes in lean mass. When we looked at the serological analysis at 8 weeks, there wasn't too much to see in terms of triglycerides and free fatty acids, and also their plasma glucose levels. But interestingly, we did see that when comparing the control and the obese exercise group, that total cholesterol levels were significantly reduced in the offspring whose mothers were exposed to the intervention strategy. Additional information that's been published also has shown that these offspring are hyperinsulinemic, those who are exposed to just maternal obesity alone, and actually that this is significantly improved with the offspring whose mothers were exercised, and so it's improving offspring, male offspring insulin sensitivity. So what impact does the maternal intervention strategy have on the cardiovascular health of the offspring? So starting off, we first showed that the offspring of the obese mothers have an increase in heart rate. And as you can see here, that this was also shown in this cohort. But what is interesting to see is that the offspring of the obese exercise group had a heart rate that was similar to controls of around 130, 135 milligrams, and this was significant. This was also the case when looking at it relative to body weight. So it's shown that the intervention strategy was capable of restoring heart rate back to control levels. So when we think about heart rate, we need to think about cardiomyocyte cell size, as the heart doesn't undergo division, it actually increases in size postnatally. So when there's an increase in heart rate, it will be likely to be due to an increase in cardiomyocyte cell size. And this is what we see in the offspring of the obese dams as their hearts are bigger. As we may expect from the fact that the hearts were restored, the offspring of the obese dams show a restoration of their cardiomyocyte cell size. And what is interesting to see is that when we look at the frequency distribution of the cardiomyocyte cell area, we can see that the offspring of the obese exercise group follow the control line. So now that we can see that heart rate is normalized and cardiomyocyte cell size has been restored as well back to control levels in the intervention group, we looked at genes that are related to pathological cardiac hypertrophy. So in the offspring of the obese dams, you can see a significant increase in some of these genes, which are known as fetal genes, and these are re-expressed under pathological conditions where the cardiac hypertrophy is irreversible. So physiological hypertrophy is something that is reversible and is often as a result of stimulus such as exercise or pregnancy. And as you can see here, that the expression of these genes here suggest that the offspring of the obese dams have this pathological form. So what is interesting to see, and probably may be expected given that the heart rate was also normalized and the cardiomyocyte cell size was also normalized, that the levels were back down to controls in the offspring from the exercise intervention group. So moving on from the cardiac cell size and the fetal gene re-expression, we next moved on to cardiac function. So previously I've shown you in the ex vivo model, so this is an isolated Langendorf model, that the offspring of the obese dams have impaired cardiac function. So using a slightly different methodology and more similar to what you would see in humans, and also this is an in vivo method, is that the offspring of the obese dams have increases in their end systolic volume and end systolic diameter, which translate essentially to a reduction in the ejection fraction. So this is the percentage of blood leaving the left ventricle during each contraction, so highlights impaired cardiac function, and also a reduced fractional shortening. When we look at the obese exercise group, however, these levels are more similar to that of the controls. So it suggests that the exercise intervention in the mother is able to restore cardiac function to control levels despite their lack of weight loss, and also the fact that they're still eating the high-fat, high-sugar diet. So in terms of additional cardiac functional measurements, we saw that cardiac output is significantly reduced, as we might expect in the offspring of the obese mothers, and actually is increased in the obese exercise group slightly more than that of the controls. And we suggest that this may be as a result of changes structurally. So here at the bottom in red, where we see an increase in the intraventricular septum and the left ventricular posterior wall, and the wall to lumen ratios increased more in the obese exercise group relative to the control and obese dams. So overall, we so far have seen that cardiac structure and function have been restored to control levels in the offspring of the obese exercise group. So next, we wanted to assess blood pressure. So as you can see here, starting off with just the two groups, the offspring of the obese dams have an increase in systolic blood pressure, suggesting kind of hypertensive phenotype, without any changes in pulse rate. We then also use the echocardiography. So here you can see the left ventricle, the aorta here, and we measured the ascending aorta diameter in systole and in diastole, and we can see that this is significantly increased in the offspring of the obese dams, and this essentially suggests to us an increase in afterload, and that is because in humans, persistent hypertension increases the width here due to the increase in afterload, and long-term hypertension and an increase here is associated with then cardiac abnormalities, cardiac dysfunction long-term. So interestingly, we see that the obese exercise group, unlike the cardiac structure and function, fit more with the blood pressure, bit more with the obese group, sorry, in terms of their blood pressure. So they don't have a restoration of blood pressure, they are still hypertensive, and they also still showing an increase in afterload with an increased width of the ascending aorta. So due to the fact that we have hypertension still remaining, we next looked at the contractile proteins such as troponin, tropomyosin, and also calcium cycling proteins such as SERCA2. So as you can see here, the obese exercise group showed a very high significant increase in all of these proteins relative to control and obese groups. So this supports a role for a potential increase or compensatory increase in these proteins, which will help to drive an improved cardiac function, despite an increase in afterload that is caused by the hypertension that we saw in the previous slide. Also, an increase in phosphorylation of troponin I indicates an increase in sympathetic activation, again, potentially a compensatory mechanism, and an increase in SERCA2, which is a calcium handling protein, allows calcium to be cycled back to the sarcoplasmic reticulum, which will help to facilitate further successful contraction. Furthermore, increases here in tropomyosin and also troponin I will help to further improve the cardiac function by improving the contractile machinery available to these offspring. So in summary, the exercise intervention is capable of rescuing the cardiac hypertrophy phenotype in offspring of obese mothers and also the dysfunction that is seen. In contrast, maternal exercise intervention was unable to improve the systolic blood pressure, and we see similar levels of blood pressure in the offspring of the obese and in the obese exercised group, and we also see the increase in afterload in these two groups. So one of the things that is important is that initially we thought that potentially the hypertension could be a driver of our cardiac hypertrophy, in addition to maybe the hyperinsulinemia that we saw previously. But what this data shows us is that this may not necessarily be the case, and the cardiac hypertrophy may not be a consequence of hypertension, or at least at 8 weeks, because we see hypertension still in the offspring of the obese exercised group, but we don't see the cardiac hypertrophy. The other important finding is that there appears to be discordant mechanisms that appear to be programming the cardiac dysfunction phenotype and the hypertensive phenotype. And this is something that we need to investigate further, as different phenotypes in the mum during the obese pregnancy are clearly responsible for doing programming these two mechanisms separately. So why really is this research important? So I've already shown you in the first slide why it's important to the mum and the long-term offspring health, but it is important in terms of public health message, because that our research has shown that despite no changes on the scales, so there was no change in the body weight due to the exercise, in the mum or in the fat mass, the exercise intervention was still very impactful on the offspring, but also on the mother. So it will help, hopefully, to encourage women that exercise is beneficial even without a change in weight. And not only will it improve the maternal metabolic health, as we saw by an improvement in both glucose tolerance and hyperinsulinemia, but it will help to prevent transmission of poor cardiometabolic health from mother to child. And also the exercise intervention strategy that we used wasn't extreme and is highly accessible to those women who otherwise wouldn't be exercising on a regular basis. So the take-home messages from this presentation are that exercising just prior to and during pregnancy can reverse some, but not all, of the adverse effects of an obese pregnancy, and that there are discordant mechanisms involved that are responsible for programming cardiac dysfunction and the hypertensive phenotype, and this is something that we are aiming to look further into. So I'd just like to finish my final slide by just saying thank you to the Cambridge Metabolic Network and the Danish Diabetes Academy for allowing me to speak and present this work today. And I want to thank everybody from the lab, but also but specifically those who have been involved in the cardiac exercise paper, and in particular Jessica Beeson, who this work formed a large part of her PhD thesis, and also would like to thank the British Heart Foundation for their funding. And thank you very much.